Okay, this is the second lecture in which we are going to learn little more about the prokaryotic cell, and the previous lecture was more about cell wall. Here we are going to read more about plasma membrane. You studied in Monera what mesosomes are to a prokaryote. But here it mentions like mesosomes acts as vesicles, mesosomes acts as tubules, mesosomes acts as lamella. These are eukaryotic structures, but in prokaryote, mesosome serves the purpose of these three, is what basically this point means. Keeping aside what context we are speaking these three structures of the cell, the student always confuses with these three terms. So I thought I'll make the differences very clear. This is a picture of the vesicles, tubules and cisternae, where you can see lamellae. Okay, first one is vesicle. Where do I come across a structure called vesicle in a prokaryotic cell or an eukaryotic cell? A vesicle is a membrane bound sac. A vesicle has a membrane, and this is a closed structure. Point 1 and point 2, they always pack fluid within them. Fluid can be water, fluid can be solution or something in a solution. They pack fluid inside. They are the transport packets. If something wants to come inside the cell, if something wants to go outside the cell usually prefers it in the form of a vesicle. And even vacuoles are vesicles. Vacuoles are vesicles. What are vesicles now? Fluid filled membrane bound sacs. What is their function? Their function is transport of materials within cell or across cell membrane. So it is a package, a packet of transport. Wherever I see such transportation happening in a membrane bound vesicle, I see them working in the cell or across the plasma membrane. Now the next one is, this is the first point, this is the second point, this is the third point, and this is the fourth point. And what are tubules? Tubules are small tubes with a lumen found in parts of cell or parts of body. you can see here, these are the tubules of endoplasmic reticulum which is being shown here, as I am seeing ribosomes. And even the vesicles have ribosomes round them. Whatever they synthesize, the vesicles will transport. And I can also see ribosomes, cisternae. Actually, all three of them can be parts of endoplasmic reticulum, can be parts of the Golgi apparatus. Cisternae means gaps. As we can see, gaps due to the folds of the tubes we call together as cisternae. But you can see the fibers which are laying down one above the other in the layer form. Whenever I see a membrane in layered form, I call it as lamellae, vesicles, tubules. So you have a better idea. I can see them as independent structures. I can see them as the modification of mesosome. I can see them in endoplasmic reticulum and Golgi apparatus of eukaryote. These are the terms you should know when you're reading cell.